Dear students, today we will discuss about cell differentiation and stem cells. Let's start from cell differentiation. Introduction. All higher organisms develop from a single cell, the fertilized ovum, which gives rise to tissues and organs. The fully developed organism consists of a number of different kinds of cells. These cells become specialized and different during the course of development. Besides its basic functions, each kind of cells has its own specialized functions, its biochemical individuality, and its morphogenetic assignments. Every cell is capable of performing the process of metabolism and possess, to a certain degree, the ability for amoeboid movement, shows irritability, and is able to react to external stimuli. However, differentiated cells are able to perform special functions or to perform them in such a manner as other cells cannot. Thus, cell differentiation is a process by which cells and other parts become different from one another and also different from what they were originally. The process of cell differentiation involves striking changes in cell shape, the appearance of new sub-cellular organelles, or rearrangement of old one, synthesis of specific products in the cytoplasm, and in some cases, their release through the plasma membrane. In molecular terms, cell differentiation means variable gene activity in different cells of the same organism, like activation of some genes and inactivation of others at the level of transcription, controlling the production of various messenger RNAs, control of translation of these mRNAs, etc. General characteristics of cell differentiation. One of the principal characteristics of cell differentiation in higher organisms is that once established, the differentiated state is very stable and can persist throughout many cell generations. Also, if once established, differentiated cells no longer needed the stimuli with which it initiates its differentiation. In many cases, before it is possible to recognize morphologically that a cell has differentiated, there is a period during which the cell is already committed to a particular change. After this determination has been made, the cell will differentiate along a specific pathway, even if several cell generations intervene before overt morphological differentiation. For example, the imaginal disc present in Drosophila embryo is predetermined to become particular type of adult structures. The development of different structures from these imaginal discs results from stepwise decisions that are genetically controlled. Role of cytoplasm of egg in cell differentiation. Many experiments prove that there is no loss of genetic information during embryonic development, and initial differences between cells reside in cytoplasm they inherit. The egg cytoplasm contains substances called cytoplasmic determinants that determine the fate of the cells. The best example of cytoplasmic determinants is given by the granules present in the germ cells. These granules are present in the vegetal poles of the eggs and have the property of inducing germ cell formation. These cells that contain these granules will eventually become the germ cells of the new organism. If the vegetal pole is irradiated with ultraviolet light, sterile but otherwise normal animals are obtained. If the UV irradiated eggs are injected with germplasm from normal eggs, fertile animals are obtained. Furthermore, if cytoplasm containing germ cell determinants is injected into the anterior part of the eggs, germ cells develop in the anterior position. As development advances, cell interaction plays a very important role in cell differentiation. At gastrulation, extensive cell movements and migrations occur, and different types of cells interact with each other, forming three primary germ layers. The fate of the cells during this stage is highly influenced by the environment of the cells. The influence of the neighboring cells on cell differentiation can sometimes be quite dramatic. For example, when teratomas are injected into genetically marked mouse blastocysts, they give patches of normal tissues in the resulting mice. The injecting cells are able to contribute to organs coming from endo, ecto, and mesoderm. Thus, through interaction with a normal embryonic environment, a normal pathway of cell differentiation was restored to these teratomas, which otherwise is undifferentiated and highly malignant cells. Molecular mechanism of cell differentiation. The gene also plays a very important role in cell differentiation. In the absence of mutation, all the cells in a multicellular organism contain the same gene. During differentiation, each cell expresses only a small percentage of the genes it contains, and cells of different tissues express different sets of genes. The differential expression of the genes can be controlled during transcription and translation. For example, transcriptional changes in hundreds of genes are necessary during the development of muscles in vertebrates. Twist protein is a transcription factor necessary to create muscle cells in flies and other animals. Twist turns on the production of other transcription factors that in turn activate genes encoding myosin, actin, and other muscle-specific proteins. Study of the expression patterns in the Twist mutant Drosophila indicate that at least 130 genes are involved in the muscle development. These genes code for proteins that are involved in determination of precursor muscle cells, that is, myoblasts, their proliferation, and in some cases, migration, and their terminal differentiation into mature muscles. For myogenic, basic helix loop helix transcription factors, MyoD, myogenin, MIF5, and MRF4, which is collectively known as muscle regulatory proteins, associate with other transcription factor and DNA binding proteins E2A and MEF to form large transcriptional complexes that drive myogenesis and expression of muscle-specific genes. Dimerization of basic helix-loop helix transcription factors with different partners regulates their binding to specific DNA regulatory sites by enhancing or inhibiting their affinity for specific sites of DNA. Besides, their accessibility to DNA is also dependent on other proteins like SY, S and F chromatin remodeling complex, histone deacetylase, and inhibitors of DNA binding proteins. The migration of myoblasts to lymph buds is induced by scatter factor, hepatocyte growth factor, and must express both PAX3 and LBX transcription factors to migrate. Terminal differentiation of muscles is under the control of activating chromatin remodeling proteins and inhibitory proteins, and do not occur until myoblast stops dividing. Thus, these muscle regulators indicate how coordinated transcription of sets of target genes and their translation produce differentiated cell types. Now, let's come to stem cells. Introduction. In multicellular organisms, cells are continually dying and replaced by new cells, but most of the specialized or differentiated cells that need continual replacement are themselves unable to divide. For example, red blood cells, the epidermal cells on the skin surface, and the absorptive and the goblet cells of the gut epithelium are terminally differentiated cells and cannot divide and replace. The lost terminally differentiated cells are replaced by cells generated from a stock of proliferating precursor cells, which themselves usually derive from a much smaller number of self-renewing stem cells. Both stem cells and proliferating precursor cells are retained in the corresponding tissues along with the differentiated cells. Stem cells are not differentiated and can divide without limit, or at least for the lifetime of the animal. When a stem cell divides, though, its daughter cell has a choice. Either it can remain a stem cell or it can embark on a course leading to terminal differentiation, usually via a series of precursor cell divisions. Thus, stem cell is a self-renewing cell that can divide symmetrically to give rise to two daughter cells with a developmental potential identical to the parental stem cell, or asymmetrically to generate one daughter stem cell and one with a more restricted developmental potential than the parental cells. Stem cells are usually present in small numbers and often have an nondescript appearance, making them difficult to identify. Properties of stem cells. Stem cells are distinguished from other cell types by two important characteristics. First, they are unspecialized cells capable of renewing themselves, though sometimes after long periods of inactivity. Second, under certain physiologic or experimental conditions, they can be induced to become tissue or organ-specific cells with special functions. In some organs, such as the gut and bone marrow, stem cells regularly divide to repair and replace worn out or damaged tissues. In other organs, such as pancreas and the heart, stem cells only divide under special conditions. Thus, self-renewal and unlimited potency are the two important properties of stem cell. In a strict sense, this makes stem cells either totipotent or pleuripotent. Multipotent and unipotent are also described to define stem cell potency. Types of stem cell. Stem cells are broadly classified into two categories: embryonic stem cells and adult stem cells. Roman number 1. Embryonic stem cells are derived from embryos. These are also known as early stem cells and found at the blastocyst stage before implantation of the embryo at the uterus. The embryonic stem cells are pluripotent and can be developed into any kind of cells of the adult body when given the sufficient and necessary stimulation for a specific cell type. The pluripotency distinguishes embryonic stem cells from multipotent cells found in adults, which can only form a limited number of different cell types. For maintaining the pluripotency of stem cells, specific genes are needed to be expressed. For example, in human embryonic stem cells, expression of OCT4 protein must be maintained at a critical level for embryonic stem cells to remain undifferentiated. Roman 2. Adult stem cells are undifferentiated cells found within the tissue of children and adults that divide to replenish dying cells and regenerate damaged tissue. They are also known as somatic stem cells. Adult stem cells typically generate the cell types of the tissue in which they reside. Adult stem cells have been identified in many organs and tissues, including brain, bone marrow, peripheral blood, blood vessels, skeletal muscle, skin, teeth, heart, gut, liver, ovarian epithelium, and testes. They are thought to reside in a specific area of each tissue called a stem cell niche. Among them, the major source of adult stem cell is bone marrow stem cells. Stem cells may remain quiescent for long periods of time until they are activated by a normal need for more cells to maintain tissues or by disease or tissue injury. The potency for embryonic stem cell and adult stem cell are different. The embryonic stem cell can be developed into any kind of cell, while the potency of the adult stem cell is limited. Besides, it is easier to culture embryonic stem cells than that of the adult stem cells. There is another kind of stem cell called induced pleuripotent stem cell. IPSCs are adult cells that have been genetically reprogrammed to an embryonic stem cell-like state by being forced to express genes and factors important for maintaining the defining properties of embryonic stem cells. Mouse IPSCs demonstrate important characteristics of pleuripotent stem cells, including expression of stem cell markers, forming tumors containing cells from all three germ layers, and being able to contribute many different tissues when injected into mouse embryos at a very early stage in development. Human IPSCs also express stem cell markers and are capable of generating cells characteristic of all three germ layers. Uses of stem cells. Embryonic stem cells have been used to study the specific signals and differentiation steps required for the development of many tissues. A more complete understanding of the genetic and molecular controls of the most serious medical conditions, such as cancer and birth defects, can be studied using stem cells and may yield information about how such diseases arise and suggest new strategies for therapy. Stem cells are also used in drug testing. The tissues regenerated from the embryonic stem cells can be used for testing the safety and efficacy of human drugs. Drugs tested on animal models may not be suitable for human beings due to species-specific differences, resulting into the entry of drugs toxic to human health. Human embryonic stem cell derived tissues and organs may be extremely valuable in identifying such drugs before they are used in clinical trials, thereby accelerating the drug discovery process and leading to safer and more effective treatments. Perhaps the most important potential application of human stem cells is the generation of cells and tissues that could be used for cell-based therapies and regenerative medicine. Stem cells directed to differentiate into specific cell types offer the possibility of a renewable source of replaced cells in the tissues to treat various diseases. Conclusion. Cell differentiation is a complex process by which cells and other parts become different from one another and also different from what they were originally. It may happen morphologically, functionally, as well as chemically. In its early stage, cytoplasmic determinants play a very important role where presence or absence of specific cytoplasmic substances induces the cells to a particular fate. Environmental factors and the cell-cell interaction also play a crucial role in cell differentiation, as shown by the development of normal tissues by teratomas when these cells are transplanted into normal tissues. Differential expression of genes that activate some and inhibits others often produces transcription factors characteristic of its fate. These transcription factors coordinately activate or repress batteries of genes that direct the differentiation process. These differentiated cells lead to terminally differentiated cells. The terminal differentiation is generally irreversible, and resulting highly differentiated cells often cannot divide. The cells in the multicellular organisms need replacement. The replacement of these cells is done by stem cells. The stem cell is a self-renewing cell that has the potential to divide symmetrically to give two daughter cells with same properties as that of the parent cell, or to divide asymmetrically to give one daughter stem cell and one with more restricted development potential than the parent cell. Generally, stem cells are of two types: embryonic stem cell with unlimited potential to develop into any kind of cells, and adult stem cell with limited potential. The embryonic stem cells are found in the blastocyst stage of the embryo, while adult stem cells are found in tissues and organs of children and adult individuals. The stem cells are useful for various purposes, such as the study of various signals and development pathways of differentiated cells and tissues, testing for the safety and efficacy of drugs, regenerative medicine, and cell-based therapies.